Good morning, friends. Today we are going to talk about the embryology. Embryology simply is made up of two words, embryo plus logy. So in this particular part, we are going to study how the development of embryo happens in the intrauterine life or intrauterus. It has been divided into certain steps like fertilization, cleavage, blastocyst formation, implantation, bilaminar embryo disc formation, gastrulation, neurulation, body folding, and further the segmentations. So it is a very big process, but we are going to study it in the bracket part, that is week wise we are going to study what is going to happen in the first week, second week, likewise. So we are going to start from the week 1, and the first process what we have is the fertilization. But for the fertilization we need egg and sperm. So fertilization is the union of egg and sperm forming a zygote with a full DNA component. Now you can see a diagram here. There is a oocyte, nucleus of secondary oocyte, first polar body. What is first polar body? These are small haploid cells formed during the meiosis due to uneven cytoplasm division. They are not fertilized and degenerate over the time. A polar body biopsy may be used as a form of pre-implantation genetic screening. So this is only for the purpose of forming of single proper cell. So cytoplasm is not divided properly. So the other one, which is small, will be degenerate. Then you can see it is covered with a covering known as zona pellucida, which is of very much importance. It is a glycoprotein coat that surrounds the oocyte, developing the embryo into the, so that it prevents polyspermy, means it allows the sperm to, one of the sperm to enter, then after that it will be not allowing anyone to enter it, it will be closed. That will be called a Jonah reaction. We'll talk about it later on. Then you can see a sperm is coming, it is hitting the wall, that is corona radiata, which are follicular cells. Then a certain process is going to happen, which will be having the acrosomal reaction. Then there will be some opening. We'll see it right now. Now, if we talk about some clinical point, the week 1 of embryonic development starts with the fertilization of egg by a sperm. This is different to how care providers count the weeks of pregnancy. Sometimes the week 1 of pregnancy starts at the first day of last menstrual period, which may be 1 or 2 weeks prior to the week 1 of the development. So there are some differences in counting the week, but the process which is going to happen will be the same like this. Next, it will be the fertilization. So how the fertilization is going to occur? So first of all, the sperm swims towards the egg and prepares for the acrosomal reaction. Then the passage of sperm through corona radiator, this will be the first step. Dispersal is due to acrosomal enzyme hyaluronidase, that is acrosomal reaction. So at the head of sperm, there is enzyme is there, that is hyaluronidase, that is which pours down and it breaks the walls of corona radiator, first of all, called as Jonah acrosomal reaction. You should always keep in the mind when the sperm was entered, it was not fully matured, and while swimming in the pathway towards the ovum, it becomes mature. That is, capacitation has happened, and after capacitation, only this process can be processed. Then you can see it will penetrate the zona pellucida, that is release of acrosomal enzyme, that is acrosin. Once the zona pellucida is penetrated by the zona reaction occurs, changes its physical property to make impermeable to the other sperm. मतलब यहाँ पे क्या होगा, एक बार sperm अंदर गया, then there will be a reaction where they are going to fill up the gap and make this wall, ये पूरा wall जो है और impermeable बना देगा, so that other sperm cannot go inside to prevent the polyspermy. In the meantime, something is going to happen in the ovum also, means the process is going to, we all know that ovum is in the second metaphase stage, which will be continued from here. It will form the different processes like we can see now. Now then there will be the fusion of plasma cell membranes of sperm and egg, which releases the head and tail to the sperm into the oocyte. The sperm plasma membrane is left behind. It will not take part in further process. Next, what will happen? The completion of second meiotic division will occur. It forms a mature oocyte and a second polar body. Again, the polar body will be discarded and the nucleus becomes female pronucleus. Next, there will be formation of male pronucleus. The tail of sperm degenerates. During the growth, male and female pronuclei replicate their own DNA. Then there will be breakage of pronuclear membranes and they will condense of the chromosome. Means both are going to move become one. Next, the mitotic cell division and first cleavage will start. Cleavage is the division of cell where only number is going to increase. The combination of 23 chromosomes from each pronucleus results in the zygote with 46 chromosomes. So this is the complete process of fertilization. So you can see there are different stages. In some books it's given there are 6 stages, in some 7. So this was the summary about the fertilization, means fusion of male and female gamete and completion of meiotic cell division also, forming the zygote with 46 chromosomes. And as soon fertilization is complete, the next process, that is first cleavage, will be starting. Next process will be cleavage. As I have told you, it is the mitotic division of zygote after fertilization, producing successively smaller cells known as blastomeres. This division occurs as developing embryo travels through the fallopian tube of the uterus. Means the movement is also happening simultaneously. ये अपनी जगह से एम्पुला से मूव करते हुए it will move towards the posterior wall of uterus. Now you can see in diagram there is zona pellucida still attached. Now zona pellucida has one more role, that पहला रोल क्या था, it is going to stop the polyspermy. Second role is it does not help to get implant anywhere. It has to travel certain distance, and after traveling the cleavage will be completed, then zona pellucida will be gone and it will implant or adhere somewhere. ये चिपक जाएगा ताकि जाके on the posterior wall. You can see polar bodies are also present here. Next, different stages of cleavage are there. It will be nearly 3 to 4 days process for traveling from the ampulla to the posterior wall of uterus. You can see now it is single cell stage, zygote. Then it will be 2 cell stage, means blastomere begins to divide. Then 4 cell stage is continue to divide, becomes smaller and smaller. Then 8 cell stage. At 8 cell stage, one more proper things is going to happen, that is known as compaction, which will make them more condensed and condensed. Next, you can see a 12 to 32 elastomeres will be formed. Or basically 16 cell stage is called as morula stage. Morula, you can simply compare this with the mulberry fruit. Mulberry like things, it's shown here. That's why it becomes the morula stage. Then what will happen? They will proliferate until it become blastocyst. Blastocyst means there will be number of cells will be there, and cyst means a cavity will be developed, which will be our next stage in the first week itself. So you can see now it comes the blastocyst formation. Step through the stages of blastocyst formation spanning from 4 to 6 days after the fertilization. You can see now cells are transformed into 2 layers. First layer, which is outermost, it is trophoblast, which is shown in yellow in color. Then you can see embryoblast, which are inner cell mass. These are called as inner cell mass. And still the zona pellucida is present. Remember, this is 4 to 6 days after the fertilization. So morula proliferates and differentiates into embryoblast and trophoblast. So what is trophoblast? Trophoblast role is very simple. It secretes early pregnancy factor and immunosuppressant protein that appears in the maternal serum. So it is the role of trophoblast. Embryoblast is everything from where now the embryo is going to be developed. Embryo plus blast meet to form embryo. Now you can see different stages. Now after that, what will happen? The trophoblast cells will cover all the boundary, periphery, and the inner cell mass will move towards the one side and get accumulated. So inner mass cell will form the embryo. Due to this shifting of the cell's position, there is a cavity is formed known as blastocyst cavity. You can see trophoblast, which will afterwards take part in the formation of placenta, and this blastocyst cavity will be a fuel field. This is full filled cavity forms, pushing the embryoblast to one pole to the trophoblast, forming the outer ring. Then what will happen? Now slowly the zona pellucida will disappear and the cells will move, already moved from one side. Zona pellucida degenerates so that blastocyst cavity increases in the size. Afterwards, you can see this is 3D scene shown to you. Blastocyst cavity, the zona pellucida fully degenerate, and now the blastocyst is ready for the implantation in the uterine wall. So this was all the things which were happening in the first week of the embryogenesis. So we'll continue for second week in the next video.